We'll look at the physical support of hereditary and, more specifically, at the cytological evidence that was used to study the transmission of chromosomes between generations. We'll talk about two sets of experiments, one done by a scientist called Walter Sutton and another done by scientists Nettie Stevens and Edmund Wilson. Mendel's work on pea plants and inheritance was widely unrecognized until the early part of the 20th century, when similar results were published by three groups of scientists whose work we will now discuss. In 1901, Thomas Montgomery studied cell division. He found that at the metaphase stage of cell division, condensed structures appeared in the cells. He called these structures chromosomes because they can be easily stained. Chromosomes can be distinguished based on their size, shape, and staining patterns. Following this, in 1903, Sutton studied spermatogenesis in grasshoppers. He used the morphological differences between chromosomes to follow their fate during mitosis, which is normal cell division, and meiosis, which is a type of cell division that leads to the reduction of genetic material. He also showed that chromosome segregation during spermatogenesis follows Mendel's laws, and later proposed, with another scientist, the chromosome theory of inheritance. Finally, in 1905, Wilson and Stevens studied gene transmission in the insect order of Hemiptera, and they were the first to associate a chromosome with a specific trait. So let's begin with the chromosome theory of inheritance that was established by Sutton and Theodore Boveri. A few fundamental points form the basis of this theory. In a given species, the number of chromosomes is constant. In a cell, chromosomes have distinct morphology, and most chromosomes have two copies, called a pair of homologous chromosomes. The number of chromosomes doubles prior to mitosis, and mitosis restores the species-specific number of chromosomes in each resulting cell. The two resulting cells receive an identical complement of chromosomes. In gametes, the number of chromosomes is halved. Each gamete contains a full complement of chromosomes, one copy of each pair of homologous chromosomes, with the exception of chromosomes that determine sex. In fertilized eggs, there is a fusion between two gametes that results in a formation of the zygote. And the zygote has a number of chromosomes that is now equal to the number of chromosomes found in somatic cells. Since half of the chromosomes that are normally found in a somatic cell will come from each of the gametes, the expected full complement of chromosomes is present in the fertilized egg. So the conclusion here is that both the number and the identity of chromosomes are preserved from one generation to the next. Chromosome assortment and segregation thus follow the laws of Mendel. Now let's look at the association between chromosomes and traits. The expectation is that if chromosomes are the cellular structures that carry hereditary information, the distribution of a given chromosome and the transmission of a specific trait must be correlated. This was demonstrated by Wilson, who performed an analysis of chromosome morphology in insects called Hemiptera, also known as true bugs, and Stevens, who worked on beetles. Let's look at the distribution of chromosomes in females. We'll start with what we call oogonia. These are somatic cells, and they will give rise to the female gamete, eggs. There are 6 pairs of homologous chromosomes, plus an additional pair of homologous chromosomes shown here in red that represent the X chromosomes. When we look at the distribution of chromosomes in the egg, we see that one copy of each pair, including the X chromosome, is present in the egg. So if we focus on the X chromosome in somatic cells, you have 2 copies of the X chromosome, and in gametes, you have 1 copy of the X chromosome. This means that only one type of gamete can be found in females. Now, what happens in males? In males, we'll look at the somatic cells that are the precursor of spermatozoids. These are called spermatogonia. Once again, there are 6 homologous pairs of chromosomes. But the difference between the male and the female is that there is no 7th pair. There is, in fact, 1 copy of the X chromosome that was present in the female, and 1 copy of a chromosome called the Y chromosome. This means that spermatogonia have 2 copies of most chromosomes, as well as a copy of an X chromosome and a copy of the Y chromosome. These spermatogonia will therefore give rise to 2 types of spermatozoid. One type, type X, will contain the X chromosome in addition to the 6 other chromosomes. The type Y spermatozoids will inherit the Y chromosomes in addition to the 6 other chromosomes. So you have one copy of each chromosome, plus X or plus Y. Therefore, in males, you have 2 types of gametes, type X and type Y. Now, if we look at a cross between a male and a female, we see that there is 1 type of egg cell and 2 types of spermatozoids, either type X or type Y. In crosses, if you have a fusion between an egg and a type X spermatozoid, then you get only females. And if you have a fusion between an X egg and a type Y spermatozoid, you get only males. So this tells you that there is a causal connection between the presence of the X and Y chromosomes and sex. And so, sex was the first trait that was associated with a chromosome. In conclusion, segregation and assortment follow the laws established by Mendel. Sex is a trait associated with the inheritance of specific chromosomes, and these observations suggest that chromosomes are the physical carriers of hereditary information.